Hello, welcome to our research project on stem cells.
In our presentation, we'll be talking about what stem cells are, the types of stem cells, the history of stem cells, some medical applications, some pros and cons, and then finally the conclusion.
I'm going to start off by talking about what stem cells are. Stem cells are a specialized cell in your body. Specialization of a cell means that the cell has a specific function to do. For example, skin cells protect your body, muscle cells are responsible for muscle construction and give you the ability to move, and nerve cells carry signal from your brain to your muscle. In contrast to the specialized cell, stem cells have no specific job assigned to them. However, stem cells are special because they have the potential to develop into any one of the many different types of cells in the body.
Stem cells are cells that are undifferentiated, meaning they do not have a specific job or function. While skin cells protect your body, muscle cells contract, and nerve cells send signals, stem cells do not have any specific structures or functions. Stem cells do have the potential to become all other kinds of cells in your body. Your body uses stem cells to replace worn out cells when they die. For example, you completely replace the lining of your intestines every 4 days. Stem cells beneath the lining of your intestines replace these cells as they wear out. Scientists hope that stem cells could be used to create a very special kind of personalized medicine, in which we could replace your own body parts with, well, your own body parts. Stem cell researchers are working hard to find ways in which to use stem cells to create new tissue to replace the parts of organs that are damaged by injury or disease. Using stem cells to replace damaged bodily tissue is called regenerative medicine.
I'm going to briefly introduce a couple types of stem cells. First, we have the embryonic stem cells, or the ESCs. Several days after conception, as an embryo begins to develop, its stem cells become pluripotent. The pluripotent embryonic stem cell cannot, under any conditions, develop into an organism. The adult stem cells are found in only specific tissues and are multipotent. Induced pluripotent stem cells, or the iPSCs, are reprogrammed adult stem cells with pluripotent properties. Hematopoietic stem cells, or HSCs. Hematopoietic stem cells are found primarily in bone marrow and are unique cells capable of self-renewal and differentiating into all types of blood cells.
Whenever there is an injury or a challenge to an organ or tissue, a whole process of renewal takes place in the body to help restore optimal health. Soon after the injury, the affected tissue releases specific compounds that travel through the blood to the bone marrow. When the compounds released by the injured tissue reach the bone marrow, these messengers tell the stem cells in the bone marrow to divide and create new stem cells, which are then released from the bone marrow into the bloodstream. Slowly, the number of stem cells increases in the blood circulation.
Now we're going to go ahead and talk about the discovery of stem cells. Early discoveries mentioning pioneering stem cell research in the 1960s by scientists Ernest McCullough and James Till. There were breakthroughs in 1998. Scientists successfully isolated human embryonic cells. And iPSC's discovery in 2012, Shinya Yamanaka and Kazutoshi Takahashi won the Nobel Prize for developing iPSCs, revolutionizing regenerative medicine.
Till McCullough's discovery of stem cells is like bang.
It was like the discovery of gravity. It led to so many different thoughts.
We had a feeling there were stem cells, to directly showing there were stem cells and be able to actually understand how those stem cells behave.
It's the biggest discovery, as fundamental as DNA by Watson and Crick.
Akin to the discovery of insulin by Banting and Best.
Their discovery of finding stem cells is like finding a new planet.
So we're going to be talking a little about leukemia specifically when it comes to stem cells. So leukemia is a type of cancer affecting blood and bone marrow, leading to the production of abnormal white blood cells. Some common treatment options include chemotherapy, radiation, and bone marrow transplants.
Nowadays, stem cell research in leukemia treatment is a very potential approaches and future direction. Stem cell research in leukemia has introduced numerous potential approaches that could significantly transform the way this disease is treated. Below are some key details about advancement in this field. First of all, is hematopoietic stem cell translate (HSCT). Hematopoietic stem cell translation is a widely used treatment for leukemia, particularly for patients who do not respond well to chemotherapy or radiation therapy. In this process, hematopoietic stem cell from a donor bone marrow, peripheral blood, umbilical cord blood are translated into patient. These cells develop into house cells relating the cancerous leukemia cells. There are two main types of translation: autologous, used for patient or stem cell, and allogeneic, using stem cell from a matched donors. The second one is the Mason's chemo stem cell (MSC) research. Mason's chemo stem cells are believed in tissue repair and immune regulation. They are being investigated as support therapy following SCT. They may help mitigate harmful immune response to ARAB versus heart disease, GVHD. Research is also explored whether MSC can reduce inflammation and promote faster recovery after leukemia treatment. And the last one treatment is chimeric antigen receptor T cell therapy, CAR-T. While not stem cell CAR T therapy involve immune cell and is often combined with hematopoietic stem cell translation. In this treatment, the beta T cells are generally engineered to carry out chimeric antigen receptor (CATR), enabling to identify and destroy cancer cells. This approach has demonstrated remarkable success, particularly in patients with relapsed acute lymphoblastic leukemia.
How does it work? So, chemotherapy or radiation is used to destroy leukemia cells and make space in the bone marrow for new stem cells. In stem cell infusion, healthy stem cells from a donor or the patient are infused into the bloodstream. Engraftment, the transplanted stem cells migrate to the bone marrow, where they begin to produce healthy blood cells, restoring normal immune function.
Tricuspid regurgitation is common amongst patients undergoing mitral valve surgery for degenerative MR. There is broad agreement that when a patient has severe tricuspid regurgitation, that valve should be repaired. However, there is significant uncertainty about how to manage moderate or less tricuspid regurgitation. And there are some data from observational studies that failure to manage less than severe TR is associated with decreased survival and heart failure.
The unidirectional flow of blood through the heart is regulated by cardiac valves that prevent blood flowing backwards. In some individuals, the tricuspid valve does not close adequately, causing backflow of blood to the right atrium. This condition is known as tricuspid regurgitation, or TR, which is associated with chronic right-sided heart failure. Although highly prevalent among heart failure patients, TR is often untreated. Patients with severe TR have reduced quality of life, as well as significantly greater hospitalization and mortality rates.
In 2013, Dr. Austin Nakano, a UCLA associate professor of molecular, cell, and developmental biology, conducted a study done in mice. From the study, he discovered that embryonic heart tubes produce some blood progenitor cells. The significance of blood progenitor cells is that they can form several different types of blood and immune cells. Ever since his discovery, Dr. Nakano and his team have been trying to figure out why the heart has the need to produce blood progenitor cells. In 2019, it was reported that Dr. Nakano conducted a new study. From the study, he discovered that blood progenitor cells generate, in fact, specialized immune cells called macrophages. He said, quote, macrophages were known to exist in heart valves, but nobody had nailed down when they arrived there and where they came from until we watched them develop in the heart tube, end quote. So why are macrophages important to the heart? Dr. Nakano said, quote, when valves are seriously damaged, they cannot be fixed. Replacement surgery is the only option. Identifying cells that contribute to valve health could reveal targets for new, less invasive therapies, end quote. Additionally, Dr. Nakano said, quote, this showed us that the macrophages that are generated in the heart tube are particularly adept at eating up excess tissue. This makes them essential not just to heart valve formation, but to heart valve maintenance throughout life, end quote.
Now we're going to talk about some of the many other applications of stem cells that have been applied in modern medicine over time. Neurodegenerative disease treatment. Stem cell therapies are being investigated to replace damaged neurons in conditions like Parkinson's disease, Alzheimer's disease, and spinal cord injuries. Organ transplantation. There is a potential to grow new organs from stem cells to address the organ donor shortage, including creating tissues for skin grafts and corneal transplants. Cardiac repair. Stem cells would be used to generate damaged heart muscle following a heart attack. Diabetic treatment. Researchers are studying the possibility of generating insulin producing cells from stem cells to treat type 1 diabetes. Now, even wound healing, stem cells can be applied to promote wound healing in burns or other injuries. And lastly, drug development and toxicity testing. Stem cells can be used to create cell cultures in lab to test new drugs for safety and efficacy before clinical trials on humans.
Some pros of stem cell research are the regenerative capabilities, the personalized medicine, drug testing and development, reduced dependency on organ donations, tissue and organ regeneration, the treatment of blood disorders, neurological disorders, diabetes treatment, and research in disease modeling.
And the next one is the con of stem cell research, the ethical concern, rate of tumor formation, immune rejection, uncertain long-term effects, expensive and complex treatment, and the last one is potential for mixes.
In conclusion, stem cells are cells with the potential to develop into many different types of cells in the body. They serve as a repair system for the body. There are two main types of stem cells: embryonic stem cells and adult stem cells. And we can see in the foreseeable future that this technology will be further advanced rapidly.